Chapter 5 Summary of Microbiology from OpenStax, which is free to everyone. Protists are a diverse, polyphyletic group of eukaryotic organisms. Protists may be unicellular or multicellular. They vary in how they get their nutrition, morphology, method of locomotion, and mode of reproduction. Important structures of protists include contractile vacuoles, cilia, flagella, pellicles, and pseudopodia. Some lack organelles such as mitochondria. Taxonomy of protists is changing rapidly as relationships are reassessed using newer techniques. The protists include important pathogens and parasites. Helminth parasites are included within the study of microbiology because they are often identified by looking for microscopic eggs and larvae. The two major groups of helminth parasites are the roundworms, Hematoda, and flatworms, platyhelminths. Nematodes are common intestinal parasites, often transmitted through undercooked foods, although they are also found in other environments. Platyhelminths include tapeworms and flukes, and they are often transmitted through undercooked meat. The fungi include diverse saprotropic eukaryotic organisms with chitin cell walls. Fungi can be unicellular or multicellular. Some, yeast, and fungal spores are microscopic, others are large and conspicuous, which means you can see them. Not hidden. Reproductive types are important in distinguishing fungal groups. Medically important species exist in the four fungal groups: Zygomycota, Ascomycota, Basidiomycota, Microsporidia. Members of Zygomycota, Ascomycota, and Basidiomycota produce deadly toxins. Important differences in fungal cells, such as ergosterols in fungal membranes, can be targets for antifungal meds, but similarities between human and fungal cells make it difficult to find targets for the meds, and the meds often have toxic adverse effects. Algae are a diverse group of photosynthetic eukaryotic protists. Algae may be unicellular or multicellular. Large multicellular algae are called seaweeds, but are not plants. They lack plant-like tissues and organs. Although algae have little pathogenicity, they may be associated with toxic algal blooms that can be harmful to aquatic life and contaminate seafood, with paralysis causing toxins. That's nice. Algae are important for producing agar, which is used as a solidifying agent in microbiological media, and carrageenan, which is used as a solidifying agent. Lichens are a symbiotic association between a fungus and an algae, or a cyanobacterium. The symbiotic association found in lichens is currently considered to be a controlled parasitism, in which the fungus benefits and the algae or cyanobacterium is harmed. Lichens are slow-growing and can live for centuries in a variety of habitats. Lichens are environmentally important, helping to create soil, providing food, and acting as indicators of air pollution. Chapter 6 Summary of Microbiology from OpenStax, which is free to everyone. Viruses are generally ultramicroscopic, typically from 20 nanometers to 900 nanometers in length. Some large viruses have been found. Virions are acellular and consist of a nucleic acid, DNA or RNA, but not both, surrounded by a protein capsid. There may also be a phospholipid membrane surrounding the capsid. Viruses are obligate intracellular parasites. Viruses are known to infect various types of cells found in plants, animals, fungi, protists, bacteria, and archaea. They typically have limited host ranges and infection-specific cell types. Viruses may have helical, polyhedral, or complex shapes. Classification of viruses is based on morphology, type of nucleic acid, host range, cell specificity, and enzymes carried within the virion. Like other diseases, viral diseases are classified using ICD-10 codes. Maybe not 10. Many viruses target specific hosts or tissues. Some may have more than one host. Many viruses follow several stages to infect hosts. These include attachment, penetration, uncoating, biosynthesis, maturation, and release. Bacteriophages have a lytic or lysogenic cycle. The lytic cycle leads to the death of the host, whereas the lysogenic cycle leads to integration of phage into the host genome. Bacteriophages inject DNA into the host cell, whereas animal viruses enter by endocytosis or membrane fusion. Animal viruses undergo latency, similar to lysogeny for a bacteriophage. The majority of the plant viruses are positive strand ssRNA and can undergo latency, chronic, or lytic infection as observed for animal viruses. The growth curve of bacteriophage populations is a one-step multiplication curve, not a sigmoidal curve, as compared to bacterial growth curves. Bacteriophages transfer genetic information between hosts using either generalized or specialized transduction. Viral cultivation requires the presence of some form of host cell, whole organism, embryo, or cell culture. Viruses can be isolated from samples by filtration. Viral filtrate is a rich source of released virions. Bacteriophages are detected by presence of clear plaques on bacterial lawn. Animal and plant viruses are detected by cytopathic effects, molecular techniques, PCR or RT-PCR, enzyme immunoassays, and serological assays, hemagglutination assay or hemagglutination inhibition assay. Other acellular agents such as viroids, virusoids, and prions also cause diseases. Viroids consist of small, naked ssRNAs that cause diseases in plants. Virusoids are ssRNAs that require other helper viruses to establish an infection. Prions are proteinaceous infectious particles that cause transmissible spongiform encephalopathies. Prions are extremely resistant to chemicals, heat, and radiation. There are no treatments for a prion infection. This was read by me, for me, so I can try to understand microbiology. Thank you.